Hi, everyone. So we're on lab 3 this week, and lab 3 is a pretty quick one. So I put the lesson and protocol into one presentation for you, and it's about estimating bacterial populations. So sometimes you need to estimate the amount of microbes in a culture tube or in a sample. And one way to do this is to use serial dilutions and plate counts. It's a technique that we use in the lab that I'm going to show you today. And serial dilutions are used to decrease the bacterial concentration to a concentration that makes it easier to count when it's plated onto an agar plate. So the concentration of microbes is measured in colony-forming units, or CFUs, per milliliter of sample. So a colony-forming unit is the unit used to measure the number of viable cells, or living cells, in a sample. And each colony that grows on your plate presumably started from one viable cell, so that's a way of counting the number of viable cells in your sample. So in this lab, you would be starting with a sample in a tube that's kind of a cloudy liquid, and it would have way too many microbes to be able to visualize and count if you were to plate it out like this. So what we need to do is to dilute that sample to a countable range. So we're going to dilute the liquid culture and then plate out the series of those dilutions to obtain a plate with between 30 to 300 colonies on the plate. So why do we want 30 to 300 colonies on a plate? Why not less? Why not more? It's because 30 to 300 is a good countable range. More than 300 would be too many to count. And less than 30 might not give you a good representation of your population. So the experimental goal is to dilute that sample enough to obtain a plate that has 30 to 300 colonies, to then count and calculate the concentration of the culture that we started with. But we don't know how concentrated the culture is that we started with. So that means we don't know how much dilution will give us that desired 30 to 300 number. So what we need to do is make a series of dilutions. So now I'll go through the protocol of how to do the serial dilutions. In the lab, each table would get 3 bottles of sterile water, each containing 99 milliliters of sterile water in the bottle, and also a broth of E. coli culture. Now remember, you would never ever shake any of these bacterial broths because that cap is not a screw top. So you would never shake it. You would only stir it gently with the loop and being very careful not to infect yourself and get any of the E. coli on yourself. So then you would follow this diagram to create a serial dilution and then add the proper volume of the diluted culture onto each appropriate plate. So starting out with your liquid E. coli culture, you're going to take 1 ml of that and put it in your first bottle of water. So you had 99 milliliters of water in that bottle. With the 1 milliliter of culture, now you have 100 milliliters of water. Then you would recap that and mix it well. Then you would take 1 milliliter from there and put it into the next bottle of 99 milliliters of water. So now you have a total of 100 milliliters here. Now you're going to mix that well and then take 1 milliliter from that and put it into the third bottle. Now you have 100 milliliters total in the third bottle. So that creates your serial dilutions. Then we're going to plate from this dilution. We're going to try 2 dilutions, plating them out and see if we can get that countable range. So you're going to plate 0.1 milliliters onto plate A from this sample, and then you're going to plate 1 milliliter from that sample onto plate B. From the third bottle, you're going to plate 0.1 milliliters onto plate C, and then plate 1 milliliter onto plate D. So it's testing some different dilutions on different plates. And remember that the goal of this is to dilute a sample enough to be able to obtain a plate that has about 30 to 300 colonies on it so that we can accurately count it and calculate the population that we started with. So here's a video showing how to do that serial dilution. You're going to start with your E. coli culture and gently mix it with your micropipetter. And take 1 milliliter of that culture and add it to the first bottle of sterile water. So there's 99 milliliters of sterile water in there, and now we've added 1 milliliter of culture for a total of 100 milliliters. And you're going to shake that and open the cap. So then, with a clean tip, you'll take 1 milliliter from that dilution and add it to your second bottle of 99 milliliters of sterile water. So you see we've diluted it further. And then you're going to close that bottle up and shake it well. And then finally take 1 milliliter from that bottle and add that to the third bottle of water. Again, 99 milliliters of water and 1 milliliter of sample. So that third bottle is even further diluted, and you'll mix that one. So each pair of students in the lab would get 4 of these Petri plate strips. And we use these in this lab instead of regular agar plates. They're a thinner material, and when you add the liquid onto them, it forms like a gel that the cells can grow in. And also it has those square grids on it, so it makes it easier to count. This is a diagram of how to use those Petri plate strips. So you would start with the strip on a level surface on your bench top, and you lift the top cover and pipette your desired volume of your sample onto the strip and let the top film lay back down onto the sample. And then it comes with a spreader. It's like a tool that you use to just press down on the sample and apply pressure onto that to distribute the sample over a circular area. This picture is showing you now the next step is plating those bottles. This is the second and third dilution bottles, so the 10 to the minus 4 and 10 to the minus 6. And it shows you the strips that you'll plate them on, different volumes onto strip A and strip B from the 10 to the minus 4 bottle, and different volumes of the 10 to the minus 6 bottle onto strip C and strip D. And this here is showing you the spreader tool that you'll press onto the sample after you put the film down. And this video is showing you how to use those Petri strips. So taking 100 microliters or 0.1 ml of the 10 to the minus 4 sample and putting it on that strip and pressing down with the tool to form a circle. And then this video is showing you for strip B, you use 1 milliliter from the 10 to the minus 4 bottle. Place that onto strip B, let the film go down, and then use that tool again to press the sample, push down and form a circle. And then you would repeat that process for strips C and D. So for strip C, you're going to use 0.1 ml from the 10 to the minus 6 bottle and use the same process to put that onto strip C. And then for strip D, you're going to use 1 ml from the 10 to the minus 6 bottle and put that onto strip D. And finally, you would place those strips in the 37-degree incubator overnight, and you're finished.